Hi, I'm Gabriella Olivier.
Hi, I'm Catherine Kunkel.
I'm Sierra Collier.
I'm Samantha Maslock.
Hello, my name is Stacy Castro, and this is Group 9 for Genetics Project.
Genes are the sequences of base pairs making up DNA that correspond to certain allele on a chromosome. Each allele corresponds to a certain phenotype that is expressed in an individual. National Human Genome Research Institute states that genes can be edited to make highly specific changes in the DNA sequence of a living organism, essentially customizing its genetic makeup. Researchers at UAB, University of Alabama Birmingham, were able to use their knowledge about gene editing to create the first clinical pig-to-human kidney transplant using a brain-dead person who fit the criteria based on a previously developed cross-match test.
According to our article, 240 Americans die every day on dialysis, which can be prevented if there's an unlimited supply of kidneys. Pigs are the most promising animal donor that can help fix the organ shortage. Researchers at UAB were able to genetically engineer pig kidneys to be compatible with the human immune system. There were 10 modified genes to ensure the xenotransplant was successful. 4 of the genes were knockouts from the donor pig, and the remaining 6 were knock-in human genes to ensure that the kidney would function properly.
A group of donor pigs lived in a pathogen-free facility. The recipient was a brain-dead human whose family consented to the procedure. His DNA was a close tissue match to the pig, and it was tested through a previously developed serum crossmatch. This crossmatch is similar to the selection of human organ donors, so viruses could not be present in either individual. After the pig's kidneys were removed, they were transferred to the UAB hospital while the recipient kidneys were being surgically removed. The pig's kidneys were attached to the recipient's arteries, veins, and bladder. Urine was produced within 23 minutes. Another huge milestone was the lack of pig cells detected in the recipient's blood, which is an early sign of rejection. But how was the kidney genetically modified?
The process of gene editing involves 3 main steps: target identification, cutting the DNA, and repair. Engineered nucleases recognize the specific pig DNA sequences that need to be altered and cut it from the DNA. The engineered nucleases usually have sticky ends that correspond to DNA fragments that are needed to be excised from the sequence and altered. This process helps target and specify the gene that needs to be modified. The sticky ends correspond to specific sequences at the ends of genes that were knocked out to ensure that they were removed correctly. CRISPR-Cas9 is a powerful gene editing tool that allows scientists to make precise changes to the DNA sequence. The CRISPR guide RNA attaches to the Cas9 enzyme and snips the DNA at the spot indicated by the RNA. For homology-directed repair, the cell's natural repair mechanisms take over and attempt to fix the broken DNA strand. Often during HDR, there is simple base deletion or insertion, causing a frameshift mutation that silences the gene. During non-homologous end joining, new template DNA can be introduced to the sequence at the specifically chosen site. This is how genes are knocked in or silenced in the editing of the pig kidney. Let's go into the specifics of the pig genes. 3 knockout genes disabled enzymes that would cause hyperacute rejection. They were GGTA1, beta-4-GAINT2, and CMAH. The 4th knockout regulated growth receptors in pigs and prevented uncontrollable growth of the kidney in its new environment. The addition of 4 human genes aimed at preventing rejection during the transplantation. Human CD55 and CD46 are complement inhibitor genes, meaning they attack foreign cells from the transplant. These genes were previously added in other primate xenografts and showed to produce positive results. Human TBM and EPCR regulate blood coagulation to prevent micro blood clots from forming on the organ and from spreading throughout the body. The final 2 knock-in genes were HO1 and CD47, which were used to reduce inflammation of the xenograft and prevent T cells from attacking the organ. The UAB study found that 1 pig kidney produced urine after reperfusion, while the other had limited urine production, but both were unable to clear creatinine from the blood. The experiment was too short to measure signs of long-term rejection since it was discontinued after 3 days, but the model is a potential solution to the shortage of kidneys for transplants since hyperacute rejection was not observed. According to the National Institute of Health, kidney transplant is a better option than dialysis, as it has a higher success rate and improves the recipient's longevity and quality of life. However, of the people who actually need an organ, 83% of them need kidneys, and only about 20% of those people will actually make it to the transplant list to receive one. Dialysis typically only works well enough for about 8 years, meaning many people die yearly just waiting for a kidney. In the future, a successful xenograft could be the key in solving the organ shortage, but this short trial was a step in the right direction. Thank y'all so much for watching our video. We hope you enjoyed it, and here are our citations.